I think we should start this next session, which is data-driven precision medicine area. And of course, one of our aims here is to generate new data and knowledge by using the data that we generate in the precision medicine domain. But I would also say that one of our big long-term aims is actually to convert this data into the patient benefits to improve the human health and precision medicine. So we have a very exciting set of speakers in this short session, and we will start by Professor Søren Brunak, who is at the University of Copenhagen, one of the founding members of the Novo Nordisk Center for Proteome Research, and of course have been pioneering the computational systems biology and precision medicine analysis, and even at the population scale. So we're very excited to hear your talk. So please, Søren. I would just also say one thing, that I'm encouraging you to ask questions in live. So raise your hand after the talk and ask questions. And if you don't dare, you can also post them into the chat. So please, Søren.
Yeah, thank you very much for the nice introduction and for the invitation. Always a pleasure. And now here to talk to you from Copenhagen. So many of you are well aware that we have worked on the disease trajectory concepts for quite some years, where we try to understand how diseases follow one another, but also how they are impacted by treatment and other exposures. And I guess exactly as you said in the introduction, I mean, we are moving a little bit from being very descriptive, because it was not really known how diseases followed one another at the population scale when you go across all diseases. But now we actually also try to make this more predictive and maybe a little bit closer to bedside, as you will see in the talk. But the main idea here is, of course, to try to understand disease progression patterns and longitudinal features in that, and to stratify patients longitudinally. That can be according to mortality or side effects or in many other ways, as you also will see in the talk. So the basic idea is quite simple, and we use population-wide Denmark data from Denmark, but these data are also present in Sweden in a very similar way. And this work started essentially with this paper from 2014, where we developed a quite simple concept to see how do we identify statistically significant disease progression patterns by seeing how diagnoses follow one another in a directional fashion. So this is not disease co-occurrence work per se, it's an element in it, but then we also go on and compute whether this particular diagnosis follows another one in a statistically significant directional way. And you can summarize this also graphically when you have these linear disease trajectories. And we've made actually a ton of papers on how to see, for example, in depression, what comes before you get your depression diagnosis and what comes after. So this is really important also in precision medicine, that we go across diseases and find out what is risk factors, what is complications, and sometimes a diagnosis can actually be both. So it's a complicated area, but as I said, we have published a number of more descriptive papers. For example, here's one from a few years ago in cancer, where we take 8 cancers and then we look what is actually statistically overrepresented if you go 10 years back. And of course, there's nothing causal in this. This is just associations, but some of it could be causal and also a starting point for example, if you have genetics data, you could do Mendelian randomization and then follow up with an analysis tools that would be pointing at more causal relationships. So in this paper, we also did some trajectories, and you see across cancers that some of these patterns can be very complicated. For example, skin cancer, you can get that in many different ways, and also some of which are provoked by treatment. So that's the complicated graph here. So we have, as I said, started to try to make use of these trajectories in a more predictive way. And here's again a paper from a few years ago on intensive care patient mortality. That was our first prediction paper where we try to predict mortality, and we use 15 years of disease trajectory to the left, and we also use acute data to the right. And the surprising result here was that the disease trajectory outperformed the data that you had 24 hours after admission, the acute data. So this was an interesting example of how informative and information-rich our disease trajectories actually are, also when we use them at sort of n equal to 1 single patient basis. And this was done before Corona, where intensive care patients were a little bit more exotic, but of course now everybody acknowledges the value of this work. So we switched to other diseases, and I'll just point at some work we are doing at the moment on pancreatic cancer. This is really a cancer that is discovered very late, and therefore the mortality is extremely high. So we've tried, in a collaboration with Presander in Boston, to try to see whether we could develop a prediction approach. And of course, when you take a disease like pancreatic cancer, where there aren't that many symptoms and diseases before, it to some extent comes a little bit out of the blue. So I wouldn't say that it's a health to disease transition, but it's not like there's been a lot of known prior diseases, maybe diabetes and so on, and a few other known risk factors. But this is really, really hard to predict. So we have a paper on bioarchives where we developed a machine learning approach to take in these disease trajectories and of course train from them and predict pancreatic cancer in the future at different time points. You can go with 3 months, 6 months, 12 months, 36 months, or 60 months, or what you like. So what we have done here, and that is also a take home from this talk, is to work with the Danish data, where we have a lot of longitudinal data, as you know, millions of patients, and in this data set we had 24,000 pancreatic cancer cases, and we had an average of 23 years of history. And we then replicated this work in Boston data with the Mass General Brigham database, where we are having fewer patients, 4,000 cases, and also fewer controls, and as you can see, 13 years of history. And that was an interesting exercise, because of course optimally you would like to take the prediction algorithm and then you will just carry it over and then use it in the same context in Boston. Of course, we knew up front that there would not be the same follow-up time. We used a sort of a deep learning approach with embeddings and encodings, and we also compared different algorithms, and I will not go into that kind of detail here. The idea is to produce an algorithm that might not perform super well, but it might be very useful in screening, where you would like to enrich for those you follow up with other techniques for scanning them and making early detection in a way where you actually have enriched the data set and the patient cohort that you screen. So one of the surprises, as we hadn't done this before, and it might be of general interest for many of you, is that I mean, in the US data, they code like crazy. I mean, in Scandinavia, when you see over to the right here, how many disease codes would we have in the cancer set and in the no cancer set. So when we look into the Danish or Scandinavian data, we would have a moderate amount of disease codes going back. But actually, when we look in the Boston data, I mean, there is a much more detail in the coding, and of course, it's related also to billing bias, and that really needs to be factored in when you try to port one algorithm from one place to another. And when we did this, we observed that it was actually quite hard to move the algorithm to Boston as it was. Here you see some areas under the curve. Of course, we also exclude some data before we predict, because in the time up to diagnosis, I mean, it becomes easier and easier to predict if you get certain scannings and so on. So we normally have a sort of a data exclusion window. But I mean, when we just take the Danish algorithm and apply it to Boston data, it has a substantially lower performance than if we simply take the concept and the algorithm and just retrain it on Boston data, and that's some problem that we have. I wouldn't really call it a problem, because it's just the fact that we cannot change the healthcare data that we have from different sites, but we need to be aware that sometimes we can port the concept and the prediction scheme, but not necessarily the algorithm in the way it is done. So this is something we work with. Of course, you can also interpret the result, and I will not really have time for which diagnosis is the algorithm paying attention to, but you can actually use this concept to spot new features that might have predictive performance for setting up such a screening algorithm that can be used in a real world trial, and this is the ambition they have in the US. So we are not planning one in Denmark, but they are trying to set up such a screening. So while we cannot really change health data across geographical locations, we quite well know that I mean, there's a lot of noise in healthcare data. When we take these disease trajectories, of course, we have mis-over and underdiagnosis happening. And what do we do about that? And I'll just mention a paper we published a few years ago, because I think it's also of general interest that we might have some disease trajectories for a given disease. If you take COPD J44 here, you might have some patients that are following trajectories which are very unusual. So you might find a lot of outliers in terms of trajectories. And I just want to mention it before I go to the exposure part of my talk. What we found here was that the very unusual COPD patients, they tended to die earlier than patients following a more usual COPD trajectory, as you can see from the survival curve here, but also these patients that died earlier, they had fewer comorbidities. It's sort of a little bit counter intuitive. So what we think is going on here, and that is, I think, of general importance when you try to clean up healthcare data, that actually some of those, in this case, very dissimilar COPD patients, some of them we think are misdiagnosed. They might have another disease actually, but also some of them might be overdiagnosed. They might not have the diagnosis at all, because some of them died fast and some of them died slower when we compared. And just to make a long story short, and the paper has been published more than a year ago, that it's interesting that when you follow up on what kind of tests the patients have had in the EHR, that some of these potentially overdiagnosed patients, they tended to have fewer lung function tests and so on. So that's one way you can sort of substantiate that the workup has maybe been less optimal. But the conclusion of this work was that we think that these patients that actually have fewer comorbidities and die faster, they are not having COPD at all, but they are actually underdiagnosed for lung cancer. So you might be overdiagnosed for one disease, COPD in this case, and underdiagnosed, and therefore also untreated for lung cancer. And it's sometimes a little bit hard to prove that patients had another disease, because they are of course dead, many of them, but you can go into the lab values, and here we could see that these unusual COPD patients that we think are misdiagnosed, they actually look more when you look at C-reactive protein, for example, like lung cancer patients. So cleaning up healthcare data in an intelligent way, I think is really also important in this longitudinal analysis of diagnosis. There's a lot of power in these trajectories, and diagnosis might appear out of context, so that we have an overdiagnosis, but also the contrary, where we have the opposite underdiagnosis. So you can play around with the Danish data in this disease trajectory browser that I will not really have time to go through, but I think that's also a trend where we sort of make tools where you can interrogate longitudinal patterns in a population, and then you can just make analysis very quickly, and you're not in need of making applications for data or moving data, but you can then compare to how the disease progression patterns look in your own population. So let me switch to what we now have started to focus much more on, namely the treatment side of things and the drug exposures of the patients. And so one problem that we of course also have in healthcare is that we have a lot of polypharmacy. People get diseases on top of each other, and we really need to do something about this and try to figure out how we deal with this over-medication situation in a context also where we are becoming older and older. So we have done essentially the same with prescriptions instead of diagnosis. So we actually analyzed 1.1 billion prescriptions from almost 24 years. And again, we go across all drugs like we went across all diseases with the disease trajectories. And you can see in the paper that was published last year. So now the color coding here is ATC groups and not ICD-10 color coding of diagnosis as before. But we get a lot of prescriptions when we are babies, and then it goes away. And then, of course, when we get older, we get more prescriptions. So we again developed an approach that will make this computationally feasible to sort of identify drug pairs where one drug would increase the likelihood of getting another drug, and then we can stitch the pairs that have a statistically significant direction into longer disease trajectories. A lot of detail in this that I will not have time to go into, of course. Also, sometimes one drug will decrease the likelihood that you get another drug, but a lot of interesting richness in these data, where you of course also can see that some drugs are generating a side effect, and then you are treated, for example, with opioids for that side effect. So there are many things you can look into when you look at these prescription trajectories. And so what came out of this was sort of a complete mapping of trajectories, prescription trajectories in the Danish population. So if we set a threshold of 1,000 patients that should follow a trajectory, we found 9 million. This is, of course, completely arbitrary. And of course, again, super descriptive and not very useful. But you can, of course, zoom in on certain drugs. And here's a metformin and diabetes relevant trajectory. But where this really becomes useful is to try to predict drug shifts. So in many different areas, you have first line treatment, you have second line treatment, and third line treatment. You start with the cheapest often. But it would be interesting to see whether these prescription trajectories actually can be predicted, because some patients should maybe start on second line treatment instead of starting on first line treatment. So this is what we go into in the paper, and this is an example for hypertension, where we compare two different types of drugs, ACE inhibitors versus ARB blockers. But you can compute survival curves here, and you can also go, for example, to another population. We also went to UK Biobank, but I skipped the details here. I mean, the point is that you can take this on to something that you potentially can use in the clinic. For example, these drug shifts, you can make a GWAS. And we have done that in the UK Biobank data, and there are also some work ongoing in Danish data, where actually the SNPs that pop up really make sense in the context of hypertension. But what we now work on is to try to make this into a decision support system, because we think we can use the previous prescription trajectory as input to a prediction algorithm, and actually a very simple one where you just look for sort of very similar prescription patterns actually might identify some of these patients that are most affected in terms of survival. But we actually think that these prescription trajectories contain a lot of information, not only on your prior diseases that you are being treated for, but also possibly of genetics. Previous work from Ross Altman's group at Stanford that looked into drug use in UK Biobank could establish that the physicians actually, they look for a drug that seems to fit your genetics, but without knowing it. So you shift patients from first line to second line and so on. But of course, in most cases, the physicians would not have access to the genotypes and the genome sequences of the individuals. But nevertheless, they try to look for something that is efficient and work, and maybe are not generating too many side effects. So in these prescription trajectories, there are presumably a lot of information about your genetics when you look at these drug shifts. So we try to make this into a decision support system, and also focus on this number needed to treat, because there's also a lot of over-treatment that maybe could be at least lowered by identifying the low-risk patients. So one thing is to analyze prescription trajectories. In a paper we have in revision at the moment. We have also looked into dosage trajectories, because the prescription trajectories that I showed before, they were sort of shift from one drug to another. We didn't look into dosage changes in that work, but we have used inpatient admission data. So this is a smaller data set. It's around 1 million patients, and you see the diseases. So the colors down here are back to the disease areas in an ICD-10, but clearly those who get many drugs, and you see the violin plots here, they tend to be older. You can clearly see that. This is not so surprising, but I mean more than 3 million admissions. So when we looked at this data set, we defined 185 million treatment episodes, and it's actually very, very complicated to keep track of all these dosage changes. Some drugs are given every other week or one-time administration and so on. So there's a lot of complication in it. Again, we broke it down into co-medication pairs, and we found nearly 4,000 pairs with significant dosage changes. So you have an index drug, then you give another drug, and then you have a statistically significant dosage change pattern. And so what do these dosage changes mean? So what we did in this paper, in addition to many other things that I have not included here, was to compare all these 4,000 pairs where we had significant statistics around the dosage changes. We could see that when we mined, we pooled a lot of drug-drug interaction databases where you have known registration of drug interactions, for example, Drug Bank or the database from the Danish Medicines Agency. So 2,300 of them for 3,300 of these 4,000 pairs, we could actually find a known drug-drug interaction. So in some sense, these dosage changes reflect to a large extent known drug-drug interactions. And then we have 17% left, and you see that to the right with this wheel here. So this was 700 co-medication pairs, and actually these patient groups, when you look, it's a little bit hard to see in the bottom here, but I mean, for these 700 unknown DDI pairs, we think these dosage changes are proxies for dosage changes. And we had lower patient volumes, and they were also, when we did text mining of full-length papers in the literature, they were mentioned less together. So we think many of these drug-drug interactions have not been discovered because the patient volumes are smaller, and it's therefore difficult to get the statistical significance when you analyze them. So it's one way maybe to spot yet unknown drug-drug interactions by analyzing dosage trajectories in the way we have done in this paper. Many more details that I will skip, but this is again an example of longitudinal analysis of healthcare data. So in my group, we are working with a lot of longitudinal data. One of the data types that we try to correlate to other data types is smart meter data. And I think I'm mentioning this because we are usually thinking of EHR data. as the sort of traditional diagnosis, procedures, lab values, prescriptions, and all that. But we will see many other data types, longitudinal data types, that will be useful for checking up on their impact on survival patterns, side effects, and so on. So we think we can take these electricity smart meter data and compute subgroups according to how early you get up in the morning. And of course, that's also problematic because you have a household with several more than one person in the household. But actually, the most common type of household, you can see that on the graph up in the right corner. It's actually singles, and we have started with these, but we haven't really finalized this analysis, but I'm just mentioning it as another type of longitudinal analysis that actually can be used. Maybe certain people should receive surgery in the afternoon and not in the morning, and so on. And we are trying to look into whether there is a correlation, for example, with complications in surgery or other side effects. So finally, I mean, as I also say often in talks, we are not having so many genes in the human genome. We are down to maybe 21,000 protein coding genes, and it's surprising. We've been involved in this Illuminating the Druggable Genome project, that 1/3 of the proteins are not very well studied, and we think that maybe many of these proteins actually relate to more than one disease, and this is of course just a postulate, but this is also interesting when we go to the molecular side of things. So I just want to mention this Illuminating the Druggable Genome project that was described in this review from a few years ago that we contributed to. So thank you again for inviting me and your attention. And here's a picture of my group and also some acknowledgements, and I'll be happy to answer questions.
Thank you very much. Such an exciting story, and I'm amazed by the length that you go to get your data, even to the electric usage in the house. So there is few questions. We can start with Teresa.
Yeah. Hello. Can you see?
Hello.
Yeah.
Yes. Yeah. Thank you very much. I am very interested in the data on the pancreatic cancer, where you said that applying the algorithm you developed using the Danish cohort, you had poorer performance in the US data. Do you think that this is only dependent on the fact that on the difference of the medical record, or can you highlight the other factors that contribute to pancreatic cancer, which are different, like a lifestyle, comorbidities, and so on?
No, you're completely right, and of course the fact that you have less follow-up time is also important here. I mean, some of these American hospitals are referral hospitals. People fly in from Florida or from Saudi Arabia or whatnot, so they are not having this life course long data collection on many of these patients, and that of course impacts. But you're completely right with obesity and other features. We just think that in terms of sort of algorithm design, it's the fine-grained coding of all kinds of things that lead to payment of the hospital. That is our primary technical problem, because I think we could correct for obesity and so on in other ways, but it's simply just another coding tradition where we also have maybe we have in Scandinavia, we have sort of split between GPs and hospitals. And that might be different in other countries, including in the US. But it's very relevant to point at these other differences. But when we talk to the clinicians in Boston in pancreatic cancer, I mean, the mortality looks very similar. You can also see that from the bioarchive space. So it's not like it's super different, and it comes out of the blue a little bit in both populations.
Yeah, I was thinking whether one can use your powerful tool, really wonderful, to see whether there are different etiology for the pancreatic cancer, because this is a problem this is one of the problems with this cancer, as you say, usually pop up.
We need to move on to the next question. I think that was a very good observation point, but let's go for Olli. Kallionen, you have a question.
Yeah, hey Søren, fantastic talk as usual. So particularly the drug use part was new to me in that sense. And I was kind of wondering that how much does it really add to your previous work on disease trajectories, these drug trajectories, and particularly I think it's a fantastic example which you show that in the ICU setting, it's the old track record that you have actually matters more. So if you add that to the drug use to it and consider other diseases beyond the ICU context, can you elaborate?
Yeah,
a bit
no, and that's a good point. And maybe it was not fully clear that the prescription trajectory data, these data are GP data. So the diagnosis registry data are hospital data or outpatient hospital data. So it's very complementary. And that's, of course, with many cancers, ovarian cancer and so on, we would really like to hook into the GP data to get more early signs. But we need to substantiate this. And of course, we are also using prescriptions in the context of pancreas cancer now to see how much can GP observations actually add, and it could also be interesting to see with the ICU patients. But I think there's a little bit of a different situation because you have some super sick people in hospital where you might not get. My intuition would be that the prescriptions wouldn't add so much to that on top of your disease history because you're old age. But we have great hopes for pancreas cancer or ovarian cancer to use these GP data more effectively. So it's more that again in Denmark we are not so good at hanging GP data together with hospital data. I think that's one of the weaknesses in our system. Now the law has been changed and we are getting new possibilities. But I think in the US they more look across some of these things but now we will try to substantiate that what is actually the predictive added value of the prescriptions in some of these cancers.
Maybe I can just ask the last question and with a relatively short answer from Søren. I think it's like a candy store for you to have all this disease trajectory data. So my question is, when you look at them, have you found that there is sort of organ vulnerabilities in individuals where you have clusters of diseases affecting same organs for sort of same people? So I'm just thinking, is there connection between that? If you get one disease in one organ, of course some of the inflammatory conditions can cause lots of other diseases, but do you see this type of organ vulnerabilities?
Yeah, this is a good question, and we haven't looked so much at that except when we went into side effects. We published a paper, I think it was in 2016, where we did text mining of some patient records and looked for side effects. And there we checked for sort of organ conservation. And the idea, this was a small data set, the idea was that we should roll this out on this large data set where we have millions of patients. And we are going to do that now. But we've really been delayed because developing these dosage trajectory models has just taken forever, and many depressed PhD students because it's much more complicated. But it's a super relevant question. We have just been a little bit set back by the complexity of some of these things when you go into for example, how drugs impact organs, both of course positively as the drug should, but also negatively when you get side effects. So it's a really good point.
Great. So thank you very much. That was really exciting data to look at the healthcare, the use of the healthcare data to generate new knowledge. So with that, we will switch to the next speaker in our session. And I'm very happy to have Peter Horvath to talk in the next talk. He is professor and director of the Institute of the Biochemistry and Biological Research Center in Hungary, but also a high-content imaging facility in Helsinki. And Peter has been doing fantastic image analysis and molecular image analysis, and I've seen you moving to actually crunch more data into the equation by adding omics data into the image analysis. So we're looking very much more forward to this talk. Please welcome.
Thank you, Janne. Thanks for the kind introduction and inviting me. You can hear me, right?
Yes, very good.
Okay, yeah. So thank you. Then I don't need to introduce myself. Let me start with a video. So this is a whole genome-wide screen. What you're gonna see here, and probably the first image-based whole genome-wide screen where we were screening for infectious diseases. And if you think about analyzing such an assay at a single cell level, then you realize that you need to build algorithms that are precise, fast, and intelligent, and the amount of data what is carried out through such an experiment is really large. For example, in this case, it was 10 terabyte and 5 billion single cell analyzed. So that brings me, what am I doing? What am I talking about today is basically my group works on image quality improvement, especially flat field correction, image segmentation, tracking and finding single cells, machine learning applications, especially profiling single cells. And finally, I'm going to show you what Janne was introducing, that we combine imaging data with molecular studies and combine single cell sequencing and proteomic measurements with single cell morphologies. And I'm going to drive it through these 4 topics quickly, starting with image improvement. So if you see a picture like this, you see couples, the beautiful C columns. Probably your eye does not recognize is that the corners of the image is much darker than the middle, because your eyes really got used to it. But in reality, actually, if we make an intensity plot, the corners are 3 times darker than the middle. We don't recognize that, but we know that it's there on your mobile phone camera and so on. It's not a surprise. It's also there on microscopy images. And you see a couple of examples here. For example, mouse kidney, yeast cells, or cancer cells. And you see the very severe effect of this so-called illumination problem in the corners of the image. And this is so severe that it can completely falsify the measurements. So we were working out a method that actually deals with that. It's called CIDR. The idea, I'm not going to go to mathematics at all, but the idea of CIDR is that we have an image, it goes through an optical system and the sensory system, and then we get the projected image, which is actually noisy, and it's because of the camera's distortion is distorted. And we said if we would have infinite number of these images and considering either of these pixels, then we should see if there is an optimal world, we should see the same distribution of the pixels. If it's not, then at least we could correct for that. And this is the idea of CIDR that was tested on different microscopy techniques. And the nice thing is that it largely outperformed basically any state-of-the-art methods and also calibration techniques. So I highly recommend to use it before you start doing your image-based measurements, and it was tested, as said, in different microscopy techniques, and you can reach it on different programming languages. So once you corrected your images, then we usually work on fluorescence images, but often also on other type of images. These images mostly fluorescent images of nuclei, some sort of particles, and the cytoplasm or other cellular compartments stained. And what I'm going to show you, it was a virus screen, an influenza screen we published a few years ago. And the major steps of image processing is detecting cells, detecting cellular compartments, and extracting their features. So basically, starting from an image, we create a segmented image where each and every cell is identified. We find the nucleus, the virus particles, the cell cytoplasm, and basically we are almost done, but we need to get some numbers. If we want to communicate that towards like a pathologist or doctors or fundamental scientists, then we need numbers out of them. And for that, we measure the properties of these cells. We measure the morphologies, like how big they are, how curvy they are, their intensities and textures. So from an image, we get a matrix where each row is one cell and each column is one property of those cells. And life is so easy, so we are done. Of course, life is not that easy in microscopy. There are very challenging tasks, like label-free microscopy, where there is no fluorescence or other stains. Cells are overlapping often, cells have certain morphologies, cells are touching each other, and this is where my group is the most interested in, and we work a lot. I just picked you a few examples in that. First one is when cells are growing on the top of each other. This often happens, for example, in cancer cells or tissue sections, that you see a couple of examples here that cells are sitting on the top of each other. So we observed one thing is that if these cells are on the top of each other, you see a brighter spot in the junction. And we said that then we could create methods that actually take that into account. And we said that, let's say a cell is something that is elliptical, and if it has a junction with another cell, then we believe that there is a brighter region in the junction. And with that, we created models, and they are actually very nice and accurately detecting the single cells, and then we can do further measurements. You can also use it for tissue images like tissue microarray spots, and you can get the single cells and then do phenotypic analysis like counting cancer cells or so on. Another thing what I wanted to show you, a very exciting, or for me very exciting, hopefully for you as well, example is when the cells are not on the top of each other but heavily touching each other. As you see here in these examples, we observed that in their contour there is a big break. It's called the curvature goes up, and then we said that probably we could create models that actually concentrate on those areas when the curvature goes up, and then they could break. So we created models that actually segment, but if there is a case, for example, we'll be here one, and there is no supporting data about the curvature up, then we just artificially move the contours so that they break up. And in this way, you can also very nicely segment the single cells. And like here, there is another of such example. And this is what we did in the last probably 10 years, and we believe that we are doing very nicely. But then in 2018, there was a world competition, the Kaggle Data Science Ball, where the task was to create software that actually can find single cells on diverse microscopy images, as you see here, several examples. And it actually turned out that this competition became very popular, and to my knowledge, probably the largest bioinformatics competition ever, with nearly 4,000 participating groups. So we said, okay, then this is our area, so let's go on and win that. And with our classical methods, we actually failed very badly, like position 1,000 or so. It turned out that everyone was using, of course, deep learning. So we also started to use deep learning for that. And with deep learning, you could be very good, like easy in the first 50 places or so. But as we also realized, deep learning is very data hungry, and in cell biology, we don't have too many data, far not as much as, for example, Facebook or Tesla would have. So it would be great to have many more data than we currently do have. And for that, we had an idea it's called image style transfer learning. Image style transfer learning is something you have a basis, a style, and then you can create that basis with that style. Or probably this picture is familiar with you. It's a German city, famous paintings, and then as if the city was painted with this by these painters. So if you can do that for cities, we thought probably we could do that for cells as well, and creating artificial training data with artificial intelligence would help. So we said that let's pick a cellular image and create another picture where we would like to have a cell here, here, and here. And we started to generate artificial images. And with that, actually, we could reach the highest score on this competition. And created this method, the nucleizer.org. You can even submit your pictures just online there, and probably it's 3 years ago now, but probably it's still the highest score, even in the post competition. And you see the results of nucleizer is quite impressive. Currently, we are working on extending this method so that machine learning learns from very few annotated examples how the structure of the data looks like and generates training data with this adversarial augmentation. I don't go too much into the details, but here I show you a couple of examples. This is a real image. This is its annotation, and from these two images, we can create a large number of artificial images that preserves the structure and the look of these cells. And we train the segmentation algorithms now almost entirely on artificial data, and they perform quite good on rather small, like 1 or 2 or 10 annotated images, which is very small in terms of deep learning. Okay, so just remember, because I made a detour, we had a fluorescent image and we got numbers out of that fluorescent image. What to do next? We would like to get some answers to the doctors. And for that, we are developing a software called the Advanced Classifier. You can find it on cellclassifier.org, where the idea is to provide very accurate analysis with minimal user interaction using advanced machine learning methods for single-cell phenotyping. Just let me show you an example here. This is a renal cancer patient-derived cell culture treated with some drugs, and we have a fluorescent stain for Ki67 marker for proliferation, and we are interested in what is the effect of the drugs on that particular patient's cells. So we tell the machine that this is a positive cell, this is a negative cell, this is a feeder cell, and by clicking it, the machine can learn and generalize to that to like any large number of data sets. And then with that, now we have a ready-made answer to the oncologist that, for example, this particular drug had this and this and this efficiency. And now I would like to show you a couple of examples where we use our methods. The first one I wanted to bring you is a childhood acute lymphoblastic leukemia screening we do together with the Children's Hospital in Zurich with Jean-Pierre Bourquin. And basically, our idea is to find the best drug treatment for this cancerous children. And the idea is that we have bone marrow cells, we treat them with drugs, put the leukemic cells on the top, and then we would like to know whether what is the effect of certain drugs. And we also do it for genes. That's why SIM screening, that's why it's written genes as well. And the difficulty here is that it's a very sensitive system and very difficult microscopy with live cells. So we basically have one color, and we need to artificially distinguish the bone marrow cells and the leukemic cells. And so like in a large picture, it looks like that. And we build machine learning methods that actually are able to distinguish the cells painted red and green. And then based on that, we can propose the treatment. And the nice thing is that now the system was applied for more than 200 children as a strategy development tool. And lastly, I would like to show you how we extended our pipelines to 3-dimensional single cell screening. So a couple of years ago, I decided that we will build an entire screening system for single-cell 3D organoids and spheroids, and I want to show you where we are with that. So first of all, as probably many of you know, screening organoids is super sensitive, and there are many factors that we need to take under control. First of all, the size and geometry of the spheroids are very important, and for that, what we do is we grow the spheroids in some sort of plates or dishes or whatsoever. Then we screen them, and we only pick those that satisfy certain morphology criteria. And for that, we built a spheroid picker that looks like that it actually screens the samples. Just one second. So it's like a small robot, screens the samples and picks the spheroids that satisfy certain criteria and puts them to the drug. So here is just 3 spheroids. Deep learning finds these spheroids, measures their properties, for example, here the area, and then comes down the robot and picks them up if they satisfy something like, for example, here one was too big. And then the robot goes there and goes and picks that one and puts to the target plate. That could be any sort of plate. All right, so once this is done, the next one what we are working on the most is how to screen large organoids. And we built a novel plate. It's just been patented a week ago that actually is able to do high content screening with light sheet microscopy. Somehow it's flickering, sorry, but we could reach a high throughput scale with 300-400 micron spheroids. And then, of course, the most difficult thing is the bioinformatics part that actually concerns with finding the single cells and phenotyping them. And for that, we built annotation tools that finds the single cells, and then you can annotate them, and also segmentation tools where you can find each and every single cell and then assign their phenotypes to that particular cell. It's a very big challenge. It's much bigger than picking them in 2D. Yeah, so yeah, this is where we are with that. And one application I wanted to show you is we do it with a children clinic and the big European Union consortium treating pediatric brain tumors. It's called the COMPASS project, where we get childhood brain tumor cells and then try to find drug treatments for the disease. And lastly, I told you that we also combine single cell morphologies and molecular studies. We built a system recently, the CAMI system, computer-aided microscopy isolation, that actually combines everything what I've shown you is correcting images, finding cells, phenotyping them with like physical extraction. So either starting from cell cultures or tissue cultures, we identify the single cells, and then we identify their phenotypes, train a machine to recognize them, and then once the machine recognized, we can say that pick me that particular cell with the laser microcapturing, and it picks up, and then we can do whatever. So first we did genomic studies, transcriptome and genome analysis. That was in this Braško et al. Paper. And now, most recently, we developed a new technology called the deep visual proteomics together with Matthias Mann and Emma Lundberg, where we start with human tissue. We identify the single cells, capture the single cells, and using ultrasensitive proteomics, we isolate those singles, the isolated single cells are measured and their proteome is measured. And I'm just very happy to tell you that the paper just got accepted a few days ago. So you can soon read it, and let me just show you a few videos where real time the machine is picking the single cells from tissue and cell cultures, as you can see here. So it's all like defined by deep learning, and the machine goes there and cuts them out, collects to like single caps, and then we can do the analysis. And lastly, just to go a little bit further more, you can isolate single cells not only from 2D samples, but also in 3D. And we actually had an idea that can we somehow pick a single cell from the human brain. We probably cannot directly, but from after operation, like the tissue kept alive, and then we built the system called the autopatcher. It's just been recently published, that actually takes images of the brain cells. It looks like spaghetti. It's very difficult because there is no fluorescence or any other cool microscopy, but we built deep learning systems to detect the single cells, and then like a 6 freedom of motion system that actually drives there the pipette and picks the single cell, or can measure electrophysiology in the cells fully automatically. So what you're gonna see here, we are like 2-3 micron deep in the brain. It was not touched by a human. The machine selected a single cell, drives the pipette there and picks the single cell, and then first measures the electrophysiology and then sucks out the content of the cytoplasm for transcriptome analysis. And with that, I would like to thank, and if you're interested, we just recently published a couple of review papers about screening, data analysis, phenotypic image analysis, 3D, and also the most recent deep learning single cell segmentation. I would like to thank my Finnish, my Hungarian group collaborators, and those who gave money to our research, and thank you very much for your attention.
Thank you very much, Peter. It was just fantastic to see the combination of the data analysis and actual engineering to generate more data. So we have time for 1 or 2 short questions. Is there any questions from the audience? You can just raise the hand. But maybe I can start. Since you have these single cell methods now, and you can and study I guess I will tell you something about new about the tissue architecture and so on. But how would you, and I like your ALL study, but how do you actually form that into the precision medicine and patient benefits? Have you sort of examples of using that?
We even have early examples, especially for complex tumors where you have several driver mutations. Then you can identify the driver sources and pick those cells out for single cell sequencing. This is something we did earlier, transcriptome analysis, and then you can do targeted combined therapies as well. So that would be the very straightforward like analysis. It's not easy, but for example, there was one lung cancer patient where we found 5 different drivers in a 200 by 200 micron area. But that was a very severe, very severe case. But this is something, for example, that you could immediately apply in cancer treatment.
Sounds very interesting. So we can pick up one more question. So Jakob, if you want to take the next question.
Hi, can you hear me?
Yeah.
Yes.
Okay. So I like this idea of using image analysis, of using data analysis to generate more data, to sort of try to learn more as we go along. And I wonder how far you could take it. For example, I mean, so could this idea for your analyses, could this idea be formalized in the framework of, for example, active learning or some other, machine learning sort of paradigm like this?
Yep.
That makes sense. Is anybody thinking about that?
Absolutely. It's an area of research that is pretty active. But it's basically, you can consider this is a clever augmentation technology or advanced augmentation technology. how far you can go, we don't really know, because the one thing very important to mention is that we are not introducing new information in the system. We just use the current system. We basically try to use it in a more efficient way. So to what extent it can go, we don't know. What we see is that really for low amount of annotated data, it can tremendously help. For large, it cannot. So it's I actually consider it as augmentation. It's not active learning. It's an augmentation technology. It's a yeah, like an advanced augmentation.
Great, thank you. I think with that we need to move on in the program, and thank Peter once again for fantastic talk. And now you have all heard about the large-scale uses of the healthcare data, and then very detailed usage of the imaging data coupled to the molecular data. And I'm very happy to present our last speaker in this session, Alberto Santos, who is actually going to talk about adding molecular phenotype data into the precision medicine equation, and particularly, I guess, proteomics data. So Alberto is working currently at the Boehringer Ingelheim, heading the data and infrastructure team at the German site for sequencing. And I believe you did most of this work at Matthias Mann's group, focusing on clinical proteomics. And for those that you have missed it, Alberto has also a paper in Nature Biotech using clinical proteomics data, very exciting recent paper a couple of weeks ago. So please, you're very welcome, Alberto.
Thank you very much, Janne. And thank you very much for the invitation. I will share my screen now. Let's see. Can you see the slides?
Yes. Very good.
Perfect. Well, again, thank you very much for the invitation, and thank you, Janne, for the reaching out. It was super nice to hear from you, and I'm really happy to be part of this comprehensive and very interesting conference. Today, I will talk about the integration and interpretation of multi-omics data using graph analytics. And as Janne mentioned, I'm currently working at Boehringer Ingelheim, but the work I will present is what we did in the Multiomics Analytics group that I led at the Center for Health Data Science at the University of Copenhagen and at the Big Data Institute in Oxford. The focus of the group was data integration of different omics modalities, and we use graphs and graph algorithms to integrate this data and graph machine learning to try to interpret it and ultimately translate it into the clinic. Why multi-omics data? Well, it has been shown that different omics technologies or integration of different omics technologies can improve accuracy in precision medicine for patient stratification, biomarker discovery, target identification, and treatment recommendation. And also, if you think about it, if you include multiple modalities, then you have a more comprehensive view of disease and disease status that can inform about the risk of having the disease or the development of the disease, and also the current status of the disease or mode of action. But translating multi-omics results into the clinic requires that those results are clear, interpretable, actionable, and quick. And you could think of biomedical blood analysis, which are basically clear, interpretable, and actionable and quick, and help the doctors to come up with a possible treatment. But when we talk about multi-omics or omics data, then there is a lack of interpretability. We have multidimensional data, and then we have to translate that into actionable results, and it's not easy sometimes, at least. So the approach that we took, it was data integration. So we have this multidimensional data, and what we do is we integrate knowledge about how the different data points are related to each other. And these relationships or knowledge comes from protein-protein interactions, pathways, functional annotations, and basically any knowledge that you can integrate either at the data point level or how different data points are related. If we look at this structure, we identify nodes and relationships, and basically that's a graph And the beauty of graphs is that they allow you to define systems of interconnected components, and they allow you to integrate heterogeneous data. And when we look at the graph, they are probably not easy to interpret, but you can then use different algorithms and graph theory to try to simplify the complexity of those structures. And one of the things that you can immediately do is to look at topological properties to define the role of some of the components, how information flows, or how the flow of information is altered when you remove a node or a relationship. So just to give an example what we mean by using knowledge to interpret omics data and what is connective knowledge, let's put as an example a proteomics experiment. In a proteomics mass spectrometry experiment, you will identify and quantify thousands of proteins, and then when you compare to your control, you will most likely end up with a list of significantly regulated proteins that it's around the hundreds. So then what we do is, let's say we have protein A, we know that it's differentially regulated, and then we start annotating this protein with all the knowledge we have. First of all, where it is expressed and can be found, which diseases are associated to this protein, and what pathways is the protein working on. And then we find evidence that support these relationships. So if we have a second protein that is also differentially regulated, and we know that it interacts with protein A, is expressed in the same tissue and annotated to the same pathways, we can start inferring some of the missing links. For instance, what are the diseases associated to protein B? And it works in the same way when we have different data types. So if we have metabolomics data, we can just incorporate this data, find the relationships to proteins, pathways, diseases, and so on, find evidence in publications that support these relationships, and then use this data to feed the prediction of new links and also improve the accuracy of these predictions. But the networks are not as simple as what you see here. Generally, you have more nodes and more relationships, but what you can do from the beginning with graph structures, you can extract information, for instance, about clusters. What nodes are connected to each other more than to the rest of the network that could be translated into functional associations. Or you can look at different measurements like centrality to define the role or relevant role of some of the nodes in this graph structure. What type of knowledge we want to integrate? So first of all, we want to integrate different experimental data, and that goes from genomics to metabolomics, but you can also incorporate any other layer information that you can find, like medical records, images, physiology, or environment. And then there is plenty of information in publications that we can extract using text mining. And we can also use the different databases, biomedical databases that are being made available, that most of them, or some of them at least, are standardized, and we can immediately use to interpret the experimental data. So with that idea, or with all those concepts in mind, we build the clinical knowledge graph. And the clinical knowledge graph is a framework to automate proteomics and metabolomics data analysis and to accelerate interpretation of this data. This is work, as I said, that we did in the multi-omics analytics, but I started, as Janne mentioned, during my postdoc years at the Novo Nordisk Foundation Center for Protein Research. And the publication link is down here, if you are interested in reading more. So in the clinical knowledge graph, we basically built a database based in the shape of a graph that is able to integrate experimental data, proteomics, and currently working on metabolomics or lipidomics, and then all the biomedical data available in databases, well-known databases, and then information extracted from publications. We standardize all this data, which allows us to then automate the analysis workflows of proteomics data. And this allows us to, first of all, the researchers are able to find the information because we have structured all this information and standardized it. They can also compare it to different experiments that have been previously integrated, and then they can focus on relationships. So they don't see protein intensities only, they see proteins and also everything else we know about those proteins and how they are related to each other. And this allows us to find hidden patterns like clusters, correlation, causality, or make recommendations. The clinical knowledge graph is an open source project. It's developed in Python, and the backend is a community version of the Neo4j database. And then we built or implemented different functionality to analyze data. And that goes into many different statistical analysis and different ways of visualizing the data. And the current status of the clinical knowledge graph, or the data model, basically, so what information we store and how this information is related, is what you see on the left-hand side. The different nodes specify the different concepts or entities that we integrate, and the links explain how these concepts are related to each other. As you can see, the protein is currently centered because we initially built a framework to interpret proteomics data. But as you can see, we relate it to diseases, complexes, biological processes, pathways, and many more information. And we also model other concepts from all different omics technologies like genes, mutations, post-translational modifications, or metabolites. The right-hand side, you can see that this is a snapshot of the graph database. And this graph database continues evolving together with the different databases that are integrated, but also with the experiments that we integrate in, and also the new ontologies or new data types that we include in there. So we started integrating many different experiments, and I think that's the beauty of it, that you can integrate all this data and then see your different experiments in the context of other experiments. And we collaborated with clinical researchers, biologists, clinicians, and network scientists, and then annotated all this data, all this experimental data, with different databases that are well known and of high quality. And we use different ontologies to standardize all the data. The workflow, when you want to analyze your experiment in the clinical knowledge graph, starts with defining a project that will provide a unique identifier, and you can use this identifier along your experiments. You load the experimental design, clinical data set, and the proteomics data set, and that will trigger an automated analysis of the data. That will generate a report with all the statistical analysis and different visualizations, but we also allow, by including Jupyter notebooks, the possibility to customize your analysis. And that's in most of the cases, you will have an automated report that shows kind of an initial view of your data, and then you will go on and customize the analysis to really translate the results into the clinical questions. And this is what you get when you get an automated report, and basically you get all the quality control plots, differential regulation, different network analysis, and also annotation of your differentially regulated proteins with these associated diseases, drugs, protein complexes, metabolites, publications, and any other information that you add to the knowledge graph. But then we have these notebooks, and we use them in two ways. One way is the recipes, which is basically showing different functionality in the clinical knowledge graph. And then we have these reports, and basically those are reanalysis of already public studies that show either that we can reproduce the results using the clinical knowledge graph, or that we can extend those results by using different analysis and annotating the results with other knowledge. Current developments, well, first of all, the addition of new data types, and specifically we are working on lipidomics data, and this is in collaboration with Carolina and Christina at the Steno Diabetes Center in Copenhagen. And this, the integration of lipidomics data brings up new challenges. It's not like proteomics data, and one of the main challenges is that the standardization of identifiers and also the annotation of the lipids. However, we are leveraging from different initiatives like lipid maps, metabolomics, workbench, HMDB, and SMPDB, which on the one hand provide an ontology to classify lipids, and they are also working on the standardization of nomenclature for lipid identifiers. And they also provide links to all the databases that will allow us to incorporate knowledge about the lipids that we identify in our experiments. This is to say that without the community and without all these resources, we wouldn't be able to really incorporate knowledge to the clinical knowledge graph. And then we are working with images, and I'm happy that Peter was presented before me, because he mentioned already the two pieces of proteomics, a combination of microscopy, artificial intelligence to identify different cell types, and then coupled with mass spectrometry. And we participated in this project in collaboration with Andreas and Fabian at the Novo Nordisk Foundation Center for Protein Research. And what we aimed to do was to visualize proteomics data on these different cells. And this again brings new challenges like single cell and single cell population data, patient heterogeneity, which goes along the question that Janne asked, and then characterization of single cells across different experiments. However, we focused on visualizing data. And here you can see this is the image converted into shapes, and then we can do different kinds of plots to understand the proteomics data in the context of the image and the spatial context, basically. And here you can see how we can map the different cells extracted from the software that Peter developed, and then convert this into an image, overlay information about either morphological features, proteomic intensities, pathways, and so on. But we are also now working on incorporating the spatial context or dimension into the graph as well. Then we have this framework, and we have this knowledge graph. We want to use it to then make some predictions, identify similarities between some of the nodes that we have in the network, and so on. And for that, we are applying machine learning on graphs using either the topological features of the nodes that we have in our graph structure, using embeddings or graph neural networks to understand disease better, drugs, and also individuals. To give an example, we already worked on a pilot project that is a graph-based definition of disease. And for that, we define different subgraphs for each disease and convert those subgraphs in a graph or node embeddings, which is basically a vector that we can then compare across diseases and build these disease similarity networks that can, for instance, be compared to the comorbidity networks or trajectories that Søren was explaining. And then we can also look at why two diseases are similar based on the connections that we use to define these vectors, and then identify confounded by indication, similar targets, and so on that could explain this similarity. We want to do the same, or sorry, just to show an example, this is a Disney plot, a map 2D representation of the vectors that we generated. And as you can see, each dot here is a disease, and then we color based on tissue-specific information that comes from the disease ontology. And as you can see, we can find clusters of tissue-specific diseases that are similar to each other, which means that we can use these embeddings to extract relevant biological knowledge. We want to do the same for subjects or individuals, build subgraphs, generate those embeddings, and then apply those embeddings to, first of all, find similarities between different patients, and then if we have treatment information, for instance, generate treatment recommendations, stratify patients, or identify targets that could be used. And also, because we are in this conference, and as I said, it's very comprehensive and there are many topics, I wanted to mention the extension of OrthoHPI. Is a database that of predictions of host parasite protein-protein interaction networks based on homology. We are updating this database using all the new versions of the databases that we use, and also incorporating more biological context. We are extending it to 24 parasite, and we are identifying species-specific processes, but also hallmarks of parasitic invasion and infection. And this is work that we are doing with the Big Data Institute and also the Tropical Medicine Institute in Medellin, Colombia. And with that, I go back to how we aim to interpret data. And so we have this multidimensional data. We incorporate knowledge, we use different algorithms to identify insights, and the ultimate goal is to identify those nodes or pieces in the network that can provide an actionable result in the clinic. With that, I would like to thank the Multiomics Analytics Group, Philip Charles, a postdoc currently at the Big Data Institute in Oxford, the Center for Health Data Science at the University of Copenhagen, the Novo Nordisk Foundation Center for Protein Research, specifically the work I did with Matthias Mann, the collaboration with the Stanford Diabetes Center and with the University of Medellin. And I'm happy to take your questions, some links if you want to contact me. And also to repeat that I'm currently working at Boehringer Ingelheim, and I will be happy to chat also about what I'm doing there. And yeah, that's
Thank you very much. Fantastic talk, really putting together all the data into the network and then starting to mine the disease relevance there. There is few questions on the chat. If Aaron or Victor, do you would like to say those questions or?
Yes, I cannot see them right now, but I'll try to find them.
In the meanwhile, I thought that your graph presentation in the end was very interesting to look at the interrelationship with the diseases, and also in cancer we do lots of these basket trials that are one mutation across all the different cancer types, and the other trial type is this umbrella trial where you try to find the best treatment for one cancer type. So I think that the adding so much data into is that do you think that you need, I mean, how much patients and cases do you need actually in order to tackle these two questions? One sort of disease type treatment recommendations, or looking at the interconnectivity of the diseases.
I think in principle we are all taught to answer that the more data the better, but I would say it depends on the quality of the data. So sometimes you don't need to integrate so much data, because if you have good quality data, then you can do better predictions. Regarding a number, I don't know. I didn't have enough time to try, or enough data in some cases. But I would love to continue doing it.
Well, I think it's like a good wine. It will just get better more you get the data. But let's go to the question. So, Victor, would you like to ask the question?
Hey, I put two questions in the chat. The first one had to do with whether you you learn your schema from the data, or you hand engineer it. It seems that you hand engineer it in one of the later slides. I saw that you're combining different ontologies. So unless you want to comment on that, I suppose that's answered. But my question, but yeah, but my second, okay, yeah.
Just a quick answer. So is the data doesn't define the model. We define the model based on the questions we want to answer.
Okay. So that's like a general methodological comment, or specific to your.
I would say it's the one that made sense to me. Yeah. Like I wouldn't include data that I'm not going to use, that will only become noise when I try to do any kind of prediction.
Okay, so you're operating under sort of like closed world assumption. You don't include all the data. You just restrict the data to the relevant parts.
Yeah, exactly.
Okay. All right. Good. My second question had to do with reproducibility of research. You mentioned that you can use your framework or pipeline. I don't know how you refer to this, both for when you're working on a paper to kind of produce your results with it, but also if a paper has already been published to kind of reanalyze it and possibly annotate it. Have you encountered cases where you actually fail to reproduce something in the context of this reproducibility crisis in bioinformatics, maybe can that be used for this?
I think in many cases, so I'm pretty sure that there are some cases like that, but I have to say the ones that I try to reproduce, and the examples are the notebooks that we generated. If the results are slightly different, it's because they use a different way of analyzing the data, or they use different cutoffs and things like this, but nothing that I found that was completely different. I found a lot of data that I was not able to integrate. That's another thing, because it was difficult to find metadata, or the data was provided in a format that was not able to. So there is a lot of manual work also in formatting the data when it is made public in repositories, because everyone is more or less providing the data in different formats, processed in different ways, and so on. So there is a little bit of work behind the scenes to get this data in shape.
So we'll finalize this session by Aaron's question. So
yeah, hi, thank you.
Hi.
So when we study at least gene-gene interaction, there are different level of certainty in how in those connections, from experimentally verified, which is obviously the most reliable category, and the smallest one, to hypothetical. And then there are different categories in the middle. I can only assume it's very much the same with proteins. How do you deal with this level of uncertainty?
So if we look at protein-protein interactions, for instance, when we integrate this data into the clinical knowledge graph, it comes from two sources: string database, which provides the scores, or BioGrid that provides curated data. So then we are taking into account these scores, these confidence scores, when we use the data to either annotate proteins or build prediction algorithms.
Even in string, you have different levels of annotations.
And you can go as deep as you want, so you can choose the evidence channel that you want to. So you can use experimental data, or incorporate text mining, or use the integrated confidence.
Yeah, thank you.
thank you. I think in the courtesy of people having time to have some lunch, I would like to thank all the speakers in this session again. It was really exciting to see maybe a next generation precision medicine under development from the data side. So with that, we'll wrap up the session, and everybody should be back and at 13:05 for the next session.